Hello everyone, I am Dr. Nidhi Bharti and I have done my PhD from CMAP JNU PhD program. Currently, I am associated with the Department of Botany at Savitribai Phule, Pune University. In this chapter, we will study about the two important plant micronutrients, manganese and zinc, which play important roles in plant growth, development and physiological processes. The learning objectives of this chapter are the role of manganese and zinc in plants, their soil availability, the modes of uptake and transport in plants, and the deficiency symptoms observed in plants. Manganese and zinc are amongst the 7 essential plant nutrient elements classified as micronutrients. Though required in small amounts, plant development requires manganese and zinc in all its growth stages. Plant development requires manganese in all growth stages. Manganese takes a significant role in various plant metabolic processes. Various plant enzymes like decarboxylases employed in the TCA cycle, numerous glycosyltransferases and RNA polymerases require manganese as a cofactor. Manganese is an obligatory element of the antioxidant enzyme superoxide dismutase MNSOD, a key antioxidant enzyme of the mitochondria. Manganese is also of importance during ATP synthesis. It is also crucial for the biosynthesis of chlorophyll and plays a role during the photolysis of water. It finds a role in biosynthesis of various plant metabolites like aromatic amino acids, flavonoids, which are an important class of secondary metabolites. Manganese also contributes in the isoprenoid biosynthesis and nitrate acetylation. Manganese shows a complex biogeochemistry in soils as it exists in numerous oxidation states, unlike the biological system where it occurs as 2, 3 and 4. The most soluble species of manganese in soil is Mn2, while Mn3 and Mn4 show slight solubility. Manganese oxides exhibit amphoteric behavior as it co-precipitates with iron oxides. Manganese dynamics in soil is driven by the soil pH. Plant uptake of manganese is dependent on the manganese oxidation state in the soil. While it can vary between a number of oxidation states, the most frequently observed forms in the biological systems are Mn2+, Mn3+, and Mn4+, where Mn4+ is the slightest stable. The extremely soluble is Mn2+, which is also the most efficiently accumulated form of manganese in plants. Soil pH is a fundamental contributing factor of manganese oxidation states in soil. Plants absorb manganese in ionic forms through their foliage as well as their root systems. Researchers have described the manganese uptake pathway as biphasic. That is, it occurs in 2 phases. The preliminary and rapid uptake phase is non-metabolic with manganese 2+ and calcium in the rhizosphere. In this part, manganese 2+ is adsorbed by the negatively charged cell wall components of the root cells. The second stage is gradual as Mn2+ is slowly exchanged. The Mn transporter families include ZIP, zinc regulated transporters, iron regulated transporter related protein, CCX, that is calcium cation exchangers, NRAMP, natural resistance associated macrophage protein, CDF-MTP, that is cation diffusion facilitator, metal tolerance protein, YSL, yellow stripe-like, P-type ATPases, and PIT, that is vacuolar iron transporter, and cation exchanger, that is CAX. Manganese deficient plants show poor development and reduced biomass. Manganese deficiency also results in reduced photosynthesis and chlorophyll content leading to intervenal chlorosis. Tissue necrosis is a distinct indicator of manganese deficient plants. Reduced lignin content is also noticed in the roots of plants deficient in manganese. Oats are predominantly susceptible to manganese deficit, which trigger off a condition which is generally identified as grey speck disease owing to the grey spots on the leaves. Zinc performs an imperative function in plant metabolism by participating in hydrogenase and carbonic anhydrase reactions. Zinc also contributes to cytochrome synthesis as well as the stabilization of ribosomes. Zinc also contributes to the cytochrome synthesis as well as the stabilization of ribosomal fractions. Zinc is essential during the synthesis of tryptophan, which is a precursor of IAA, a form of auxin, which also takes a part in auxin production. Zinc is vital for maintaining the cellular membrane integrity and thereby preserving the structural orientation of macromolecules and the ion transport systems. Soils primarily receive trace elements like zinc mainly through geochemical and pedochemical weathering processes of the parent rocks. Apart from the weathering process of the parent rock material, the type, intensity of weathering, climate, and several other important factors prevail during the progression of soil formation in determining the total soil zinc content. High pH and higher calcium carbonate, organic matter, clay, and phosphate can improve zinc condition and its availability in soil. Epidermal root cells possessing root hairs take up water and zinc from the soil. These elements are then transported centrifugally in the direction of the xylem from the epidermis, which are further distributed to stem and leaves. Zinc is transported across various membrane barriers by involving transport proteins which are specialized in zinc transport across various membrane barriers is performed through specialized transporter proteins. The most well-known families of these transporter proteins are ZIP, ZRT, IRT-like proteins, HMA, that is heavy metal ATPases, and MTP, metal tolerance protein, or CDF, that is cation diffusion facilitator. The primary reason of deficiency of zinc in soil is its pH and the redox potential of the soil. Plants deficient in zinc show characteristic features. The symptoms, however, initially appear on young leaves. The most common zinc deficiency symptoms are stunted growth, reduced internodes, petioles, and small deformed leaves, the condition also known as little leaf, resulting in a rosette formation. The stems in monocots show characteristic fan-shaped leaves during zinc deficiency. The most observed symptoms in rice are wilting caused by the reduction of turgidity in the leaves, basal chlorosis of the leaves, and the late development of the plants. Bronzing is a characteristic deficiency symptom observed in the rice plants. The zinc deficiency can also lead to the death of rice seedlings during the growing stages. The chapter can be summarized as follows. Manganese and zinc play an important role in plant growth and development. They are essential components of various plant enzymes and play an important role in plant physiological processes. Their availability in soil is subjected to their oxidation states and the soil pH. Specific transporters are present in the plants for uptake and transport of manganese and zinc in the plants. Characteristic deficiency symptoms have been observed in plants deficient in manganese and zinc.